In this video, I'm going to go through the knowledge and technique that you need to answer the kind of question that you could reasonably expect in your Oxbridge bioscience interview. If you're planning to study natural sciences, biochemistry, biology, or biomedical sciences, then this is for you. Let's get straight into it. Elon Musk is a really strange man. Only 2% of his DNA is protein coding. To what extent are these statements true? Let's look at the first one. So, his strangeness is mainly due to environment. The key with this question is to understand what the question is actually looking for. And what we're talking about here is environment versus genetics. We also want to talk about polygenic traits. So, this is the idea that complex phenotypes like height, weight, intelligence, creativity are as a result of multiple genes coming together as well as environmental factors. And for the medics in the room, you might want to comment on some of the anatomical features that we can see. So, it's really important to make full use of the diagram. So, you might want to point out vitamin D deficiency, protruding chest. The key here is to use the information fully. So you want to pick out the keywords in the question and also respond to any visual information that you're given. Let's take a look at the second statement. The other 90% of his DNA is junk. So we can add in some normal genes and he will be fine. Let's break down all of the relevant components of this question. So there are a number of things we want to address in this question. The first thing that I would start with is actually defining what we mean by protein coding. So you want to actually talk about defining a gene, and things you might want to talk about within that include a promoter, allows the gene to be recognized by RNA polymerase. You want to maybe briefly outline the transcription to translation process, but obviously don't elaborate too much on that because it's kind of basic GCSE A-level knowledge. You then want to address, basically challenge what is meant by junk DNA. So obviously this is a slightly provocative statement. A lot of this DNA isn't junk. You actually want to say what it is. So this will include things like the telomeres, things like centromeres, and it will also include viral sequences that have been integrated into the human genome over time. These are mainly what we would call repeated sequences. The other element of the DNA is you want to talk about regulatory elements, enhancers. Now, the final thing that you want to address with a question like this is adding in genes. You want to comment on how you would actually technically do that. So you need to be thinking about a vector for actually getting the genes in. Okay, you want to think if this is going to be in vivo or ex vivo. So, for example, ex vivo would be when you take immune cells out and you modify them genetically in the lab, then put them back into the patient. So, in summary, you want to address all parts of the question, define key words when necessary, and effectively look at the question behind the question. Let's take a look at the final sub-question. How would you identify what genes are the ones contributing to Musk's unique brand of crazy genius? Let's break this down. The two key phrases that I'm looking at in the question are identify genes and unique brand of crazy genius. The word identify genes implies some kind of DNA sequencing. So, how might we go about obtaining this DNA sample? One way is to be a financial journalist and maybe ask him a question insinuating that SpaceX is a massive scam. As a result, he may then proceed to spit in your face. Also, we might want to look at the epigenome sequencing. We might want to see if he has different methylation or acetylation markers that regulate the actual expression of those genes. Similarly, we might also want to look at the RNA-ome, i.e., the actual levels of the various transcripts, the quantity. The other important thing to know is, what are we actually looking for? The human genome is very, very large. And in order to identify what Musk has different from the general population, you need a reference. That reference is the Human Genome Project, whereby the human genome has already been sequenced and each sequence is known to correspond to a particular gene. You're going to be looking primarily within genes, and that's very easy to identify within a computational database because you can just search for promoters. Now, if we're focusing on his unique brand of crazy genius, we might be interested in genes to do with transmitter production, neurotransmitter breakdown, the actual receptors for the neurotransmitters, anything that could suggest a slightly altered neurochemistry. However, you might want to look outside the obvious candidates for neurotransmitter related genes. One way to do this is by an analysis of SNPs, single nucleotide polymorphisms. To analyze SNPs, you need to do genome-wide association studies. The way these genome-wide association studies work is that you need a large sample for both your group of interest and your control group. So you need to get the DNA of about 1,000 of these eclectic geniuses and then have a similar control group. And then you would essentially compare the frequency of the variants at each SNP position. So, for example, let's say at position 1533 on chromosome 1, if you notice that in the Musk type genius group, 75% have the nucleotide A versus in the control group, only 21% have nucleotide A, you might statistically suggest that A has some contribution to the phenotype observed in that group. Of course, this is all inference. This is all statistical. It's not direct evidence. But as I said at the start of the video, complex phenotypes are due to polygenic inheritance. So multiple of these SNPs will be working together to contribute to that phenotype, as well as, of course, environmental factors. If you found this video useful and would like further support, you can find me at www.elvisoxfordcoach.com. In this section, I have a series of blog posts which go through the whole Oxford and Cambridge application process, covering course choice selection, personal statement development, and interview technique with examples of solved interview questions. If you would like further one-to-one support, please feel free to get in touch for a free consultation and we can set up something specific towards your needs.